So Elicera Therapeutics is a cell and gene therapy company with roots from Uppsala University. You see two of the co-founders here, Professor Magnus Sand and his research colleague DU. They have, together with their research team, developed two CAR T-cell therapies, two oncolytic virus therapies, and also one universal CAR T-cell arming technology we call iTANK. We published preclinical proof of concept data for the iTANK technology in Nature Biomedical Engineering last year. And this year we published preclinical data for our solid tumor CAR T program in GBM in Nature Communications. The company is listed on Nasdaq First North. We have a market cap of around 100 million today, and we have cash runway that lasts 2024 out. This is a pipeline picture. The iTANK platform, you can say, is the backbone of most of our programs. So we have the iTANK arming in 301, 401, 201, but not in ELC100, which is an oncolytic virus currently being developed in neuroendocrine tumors. Taking them one by one, ELC301 is our CAR T program in B-cell lymphoma. We received a large grant from the EIC Accelerator Fund of 2.5 million euros last year to support an upcoming clinical trial. So we have already received a conditional approval from the Medical Projects Agency to start a trial in B-cell lymphoma, and we expect to treat the first patient in the beginning of next year. ELC401 we've developed against a target that is overexpressed in a number of solid tumors, but initially we have developed this specifically for glioblastoma. ELC201 is what we call a next-generation oncolytic virus. It's armed with the iTANK platform, but also with an additional immune-stimulating factor to really kickstart a bystander immune response. And ELC100, even though it doesn't contain the iTANK arming, we have developed this oncolytic virus specifically for treatment of neuroendocrine tumors. We have an ongoing clinical study, a dose escalation trial in 12 patients. We've treated 11 out of 12 patients, so we have one patient left to treat before we can close this part of the study. And we expect to recruit the last remaining patient, hopefully during this year. Now, talking about CAR T-cell therapies, what is it? So CAR T-cell therapy, you can say, is a new type of immuno-oncology. So immuno-oncology is a vast space of different methods and therapies to use the patient's own immune system to treat cancer, basically. And CAR T-cell therapies have been shown to be very promising, especially in the treatment of different types of blood cancer. So what CAR T-cells can do is that they can efficiently identify and kill cancer cells, and they can also survive in the body for long periods of time, helping to counteract recurrences and metastasis, which is, of course, a big problem in oncology. So what you do normally in CAR T-cell therapy is that you extract T-cells from the patient. You genetically modify them so that they express a chimeric antigen receptor or a CAR molecule on its surface. You expand them in test tube and you re-inject them into the patient. And the idea is then that this CAR molecule will recognize a certain target on the tumor cell, so they will scan the body and they will target all of the cells that carry this specific target. This has worked extremely well in different types of hematological or different types of blood cancer. You see the first patient that was treated with a CAR T-cell therapy and was followed up last year after 10 years of having underwent the treatment, and he was tumor-free and he had an active immune system against cancer. The same situation for the child that you see here in the right corner, also had a diagnosis that wasn't able to treat with any other treatments. She got a CAR T-cell therapy, and 10 years later she was still tumor-free, considered cured with an active immune system against cancer. And this shows you the great potential that these types of therapies have. But so far, you only have approved CAR T-cell therapies, 6 different ones, in hematological cancers. And obviously, the biggest unmet medical need and the biggest proportion of cancers belong to solid tumors, 90%. So this is really where we need to make some new advancements in order to get these therapies approved also in this area. And this is what we believe that we can help to do with our iTANK platform, and I will describe how shortly. You can see from this slide also that most of the programs under development globally are still in preclinical phase. A lot of them are in phase 1, but relatively few are in phase 2 and onwards. Meaning that even though there are 6 approved CAR T-cell therapies, this is still what you can consider a early development for these types of therapies. And this also gives Elicera a lot of partnering opportunities, since the iTANK platform basically could be used to arm any CAR T-cell under development. And the earlier we can come in to offer these to other CAR T-cell developers, the better. So how come this works in blood cancer but not in solid tumors? Well, there are 2 major challenges that CAR T-cell therapies face in treatment of solid tumors that they don't face in treatment of blood cancer. In solid tumors, it's very difficult to find a target that is overexpressed on all tumor cells. So the CAR T-cells will effectively kill all the cells that carry these antigen targets. But if a tumor cell doesn't carry the antigen target, it will not be attacked by the CAR T-cell. It will form a metastasis that isn't able to treat with CAR T-cell therapies. This is a major problem you have in solid tumors, which you don't have in blood cancer. Another major issue is that in a solid tumor situation, you have a very immunosuppressive tumor microenvironment that directly suppresses the immune system and counteracts the effectiveness of the CAR T-cells. You don't have this issue in blood cancer. Also, looking at B-cell lymphoma, which is considered a blood cancer, even though CAR T-cells are very effective in this indication, about half of the patients relapse, and when they do, they often lack the target antigen, meaning that they can't continue to be treated with CAR T-cell therapies. So the iTANK technology is really what differentiates Elicera from other companies in this space. You can say that CAR T-cell therapies have been developed in different generations. First generation were shown to be quite poor, so they were improved in second, third, and then fourth generation. And what differs fourth generation CAR T-cells from the other ones is that a fourth generation CAR T-cell contains a genetic modification. You basically insert a transgene that codes for an immune stimulating substance. Usually it's a cytokine, and usually you only use one, in some cases 2. And this can trigger the immune system. The difference with what we're doing with the iTANK platform is that we are not coding for a cytokine. We're coding for a bacterial-derived protein from Helicobacter pylori. This is a neutrophil activating protein, which we short is NAP here. So what happens when this bacterial-derived protein is released is that it will kickstart a pro-inflammatory reaction that contains over 20 different immune-stimulating substances. And we believe that these many different substances together can collaborate to more optimally activate the immune system against cancer. So we published preclinical proof of concept data for this iTANK platform in Nature Biomedical Engineering last year. There we could show a broad attack on multiple targets. We can show that we can create a, so to speak, immunologically cold tumor into an immunologically hot tumor, which is exactly what you're after in immuno-oncology. We can show more efficient and durable responses for our iTANK-armed CAR T-cells when treating mice in comparison to conventional unarmed CAR T-cells. And very importantly also, adding the iTANK platform doesn't add any toxicity, so it doesn't make these therapies any worse, so to speak. And we also have data indicating that this is universally applicable to any CAR T-cell. So it's agnostic to what target you have or what other modifications you've done to your CAR T-cell therapy. So this is what this slide is trying to highlight here, that we can use this platform and offer it to any CAR T company out there developing their own programs. You can just add this and make them basically next fourth generation CAR T cells. So the iTANK business model is to license this on a non-exclusive basis and or enter co-development partnerships with other CAR T developers. And this also means that we have what we call a hybrid business model for Elicera, where we, on the one hand, develop in-house drug development programs, where we invest substantial resources into these programs with the aim to ultimately license them after hopefully having reached clinical proof of concept. In some cases, we're open to do that in preclinical phase. But the point is that these programs, we aim to generate higher revenue streams, higher upfront payments than you could perhaps expect from an iTANK licensing. However, the iTANK licensing, there we expect to have recurrent revenue streams. So we expect to be able to license this many times to different CAR-T developers. The study that we have planned in our first CAR-T study is in B-cell lymphoma. We call it the KarMMa study. It's in total of 18 patients we will treat. First 12 patients will be in a dose escalation phase, the last 6 in maximum tolerable dose. We expect to treat the first patient in the beginning of next year and have the first data report from the first 12 patients in the second half of 2025. And we expect this to be a significant value inflection point for Elicera and also something that will help us in our partnering discussions. And also the ongoing clinical trial that we have in neuroendocrine tumors with our oncolytic virus. It's a study that is supported financially in full by a neuroendocrine tumor foundation, so it doesn't cost Elicera anything. It was set up by Vince Hamilton, who you see here in a picture, and his family, who is running the foundation today. And as I mentioned before, we've treated 11 out of 12 patients. We haven't seen any adverse effects, and we've actually seen some signs of promising clinical activity already in this dose escalation trial. And we have an approval to move into phase 2 to treat an additional 12 patients with maximum tolerable dose. I think I've overspent my time here, so I will say thank you so much for listening and open up for questions.
Thank you, Jamal. Well, we have a couple of questions for you here. One is about the general field of cancer therapy, stating that within cancer therapy, immunotherapy seems to be at the forefront of innovation. You could take issue with that if you want, I guess. Is cell and gene therapy going to disrupt that perception?
I think there's a lot of expectations on cell and gene therapy because you have many different opportunities to use that type of therapy to treat cancer. And we're excited to be developing 2 different types of therapies here, both oncolytic viruses and CAR-T cell therapies. We have a little bit more focus on the CAR-T cells, that side today. But I really hope that today I've been able to clarify and really show that the iTANK platform is what we consider the backbone of our different programs here.
There's also a question asking you to elaborate a little bit on both the challenges and the possibilities of working in such a new treatment field.
I mean, I would say that the challenge is that this is quite unique. It's always good if you can show other examples of similar technologies. It's always easier to convince potential partners of the value of it. We're very happy to have published the preclinical proof of concept data in Nature Biomedical Engineering last year. That opens up a lot of doors for us. It shows us that the level of science is on a quite high level. But this is quite unique. So we've actually only found one other company that developed something similar, a Japanese company that has a similar platform that they've been able to license on multiple occasions on a non-exclusive basis. So we hope to be able to replicate that and have the same model.
And there's also a question about if there's anything similar out there that's being used for already approved drugs, but there's...
Not for the approved ones. So the majority of CAR-T cells under development today belong to 2nd generation. Very few are being developed as 4th generation, and we believe that most of the ones that are being developed as 4th generation are being developed in academia. Very few companies that we know of have a similar technology.
How come?
It's a field where you constantly learn, and you constantly learn from others, and this field is evolving, and I think more and more people will look into these types of technologies. So I expect that, of course, we will not be the only ones doing something along these lines going forward. But right now, we believe that we are in the forefront in this specific area of arming CAR-T cells, and we hope to be able to capitalize on that.
And as a final question then, do you only want to focus on solid tumors, or will you continue in the field of blood cancers as well?
So we're targeting CD20 in B-cell lymphoma, and B-cell lymphoma in itself contains different subgroups of patients. There might be a plan, depending on what data we gather from the upcoming trial, where we choose maybe to focus on a specific subgroup. But it's difficult to say that now. But besides that, no, I think that the solid tumor area is more interesting. We will start with glioblastoma, aggressive forward brain cancer, but this target that we know is overexpressed in glioblastoma, we also believe is overexpressed in a number of other solid tumors. So down the line, there could be an opportunity to broaden the pipeline also within that area. Yes.
Well, thank you so much, Jamal.
Thank you. Thank you.